My name is Julia Bergia, and I'm a junior at Detroit Country Day School. Over the past 3 years, I've dedicated a lot of my time to science and scientific research. I've done projects on the genetics behind Alzheimer's, changes in water quality variables with precipitation, and optimizing nutrition standards for competitive rowers. Although these topics may seem incredibly disconnected at first, I've begun to see that eventually all branches of science end up fitting together like puzzle pieces, at least when you zoom out far enough. But every once in a while, you finish a puzzle and end up with a piece left over, and that's the situation scientists have found themselves in after discovering a cellular organelle called the volt. In 1986, Leonard Rome and his colleagues were trying to isolate cellular vesicles in a lab at UCLA when they found something contaminating their samples. It looked a little something like this. The contaminant was a particle, about 3 times the size of a ribosome, made of proteins and RNA. This particle turned out to be a newly discovered cellular organelle, which Rome's team named the volt, because its shape resembled the vaulted ceilings on cathedrals. Rome and his team quickly shifted their focus to vaults and began to do more research. They found that vaults are highly conserved in eukaryotes, which means that they're found in most eukaryotic organisms, with very few differences between species in terms of their structure, function, or genetic code. However, they are notably absent in fungi, plants, and organisms like yeast and flies. In addition to being highly conserved, it also turns out that vaults have very simple structures. They're made up of only 3 different proteins and 1 very short type of RNA. The outer shell is made of 78 copies of a major vault protein, or MVP, which attach to one another in a circular pattern to form the outer shell of the vault. MVP is highlighted in red on this picture, and you can see how the different strands interact with each other to form a shell. The inside of the vault is made out of 3 components. The first is vault RNA, or vRNA, at the top and bottom of the structure, which interacts with telomerase-associated protein 1, or TEP1. Vault poly-ADP ribose polymerase, or VPARP, is a circular protein that binds to the inside of the MVP proteins, stabilizing the structure, which finalizes the shape of the organelle. The vault's top and bottom halves can come apart at acidic pHs or at high temperatures, releasing the internal contents of the vault. Despite knowing a lot about its structure, scientists are still unsure of what the function of the vault is. Multiple studies have been done about the role of vaults in multi-drug resistance and in tumor development. Scientists have found that high MVP expression is typically correlated with aggressive cancer tumors and poorer outcomes, which indicates that MVP might play a role in drug resistance. MVP also regulates cellular pathways concerning cell survival, growth, and apoptosis, or cell death, all of which are incredibly relevant to the study and treatment of cancer. However, a key study done by Marieke Mossink and her group of researchers suggests that vaults and the major vault protein may not play as significant a role in multi-drug resistance as some scientists may think. In the monumental 2002 study, a group of researchers removed the gene coding for the major vault protein, MVP, from a group of mice. Not only were the mice completely fine, but they also didn't exhibit a higher or lower resistance to cytostatic agents or the drugs that the scientists tested them with. This study indicates that vaults are not necessary for eukaryotic life, nor do they play a significant role in drug resistance. This discovery has stumped scientists for more than 20 years and is still being cited by research teams to this day. However, just because scientists can't seem to figure out what vaults do, doesn't mean they can't try to put them to good use. In 2001, Rome, the vault guy, decided to try implementing an MVP gene from rats into moth cells. Moths are insects, so they don't produce vaults or any of their components naturally, but after receiving the MVP gene, they were able to produce the major vault protein, despite the fact that they themselves did not produce vaults. Even more interesting was the fact that the moth's MVP proteins assembled into normal functional vaults, even without vRNA, VPARP, or the TAP1 genes. This monumental discovery gave scientists a way to create simple, synthetic vaults and sparked countless research projects about how vaults can be used in medicine. Because they're made of proteins, vaults act as perfect nanocontainers for a variety of molecules, including drugs and enzymes. Rome is currently researching how vaults can be used in cancer therapy to deliver CCL21 to cells. CCL21 is a signaling protein that starts an anti-tumor response in cells, helping our bodies fight cancer. This therapy usually requires repeated injections of CCL21 into cells, which is inefficient and definitely uncomfortable. Rome is raising money to start a project that tests whether vaults can be used to enclose CCL21. He thinks that vaults might be able to carry this protein into a patient's body and slowly release it into their cells, eliminating the need for injections and making the treatment much smoother and more comfortable. Vaults also have the potential to solve one of the biggest problems in gene therapy. The development of these therapies has been one of the most influential discoveries of modern medicine. Through this treatment, scientists use viruses to transport pieces of DNA or RNA into cells, which can then be used to make proteins, helping people with genetic mutations manage their symptoms. However, using viruses as transport vehicles presents some issues, mostly because our bodies have gotten pretty good at defending ourselves against them and can't always tell the good viruses from the bad viruses. David Curiel, a gene therapy researcher at Washington University, thinks that putting these viruses inside volts may solve the problem. Because volts are naturally made in our bodies, they're unlikely to trigger immune responses the same way that viruses do. By hiding viruses from our immune system in volts, researchers might be able to safely deposit life-saving genetic material directly into our cells without triggering an immune response. So, vaults could be revolutionary in the field of medicine, but that's not all they're being limited to. Vaults are also being tested in the context of environmental science. Some experiments were done with synthetic vaults to test whether they can control the release of enzymes that can help break down groundwater pollutants. A research team, led by Anjali Loth, showed that using vaults as transports actually helps stabilize these enzymes, allowing them to work faster and more efficiently in cleaning up our groundwater. I've done a bit of my own research testing water quality variables in a river near my house, so to hear that a cellular organelle that was accidentally discovered almost 40 years ago is now being used to improve water quality was incredibly interesting. In conclusion, vaults' ability to act as biological nanocontainers means that they can be applied to a wide range of scientific problems, both biological and not. Vaults are still a scientific mystery, but they have the potential to be an incredible success story. Sometimes that last remaining puzzle piece can be the key to a whole new world of possibilities.